I'm profusely elated to introduce our next keynote speaker, Dr. Touqeer Ahmed, Applied Bioscience, NUST, Islamabad, to share great knowledge with us. Sir, Mike awaits you. Please put your hands together for Dr. Touqeer Ahmed.
Thank you so much, organizers, especially Dr. and the whole team for inviting me and the whole team of NUST over here. And it's really nice to see people face to face after COVID, because during the COVID time, like all the conferences, they were gone offline, like online ASAB, we were sitting on the computer all day. And like being present here and talking while you have the audience in front is obviously, like it has a very different feeling. So I will not take much time, and it has been a great learning day. Dr. Ternam was my teacher, was my co-supervisor, and some of the people like who gave talk, they were like also, they have also like played the made the ground for me, because I will talk about amygdala. And today's talk is mapping the unmapped connections, mysterious puzzle for setting memory strength. We have been listening about the formation of memory, neurobiology of the memory. Dr. Ternam told about the amygdala. So I will talk about how a strong memory and a weak memory is formed. And particularly in those different types of memories, how aversive memories strength is defined. I will touch on a circuit that actually starts from the amygdala to the periapical brain area, which has an important role in pain, and rostroventral-medial medulla, which is part of the brainstem that we have already heard in the talk of Dr. Ternam. So this is my location where I am located in NUST. The neurobiology lab is located in this building. This is a bit old picture of NUST, but just to give you an idea, it's a big setup. Here is the rector office, principal office is here, and across the road is animal lab where we do a lot of behaviors and then do analysis in this building. There is Power School of Applied Biosciences. So how, the question is what is the strength of memory? How memory strength is made, how it is formed, and especially what are the aversive memory strength, how that is actually defined, made, where the amygdala plays key role, and in the amygdala is responsible for the fearful memories. When, for example, if I see a snake here or any other fearful event happening in my life, and that then basically amygdala gets hyperactive and then results in the formation of associative memories in the brain. Like you see a snake here, or the spider-like tissue, which they show a fearful response. Fearful response is usually a survival response, because if you do not present fear, you can do something which can be fatal. So if we see the structure of amygdala, so this line on the lower left side we see the section of rat brain, and this line is showing you a histological section of the amygdala where there is central amygdala, lateral amygdala, basomedial amygdala, and there are further subdivided, and people have spent more life elaborating the role of amygdala, especially if we talk about Jolie Doe, who has been the father of this brain area, UPI, and he has now a music group with the name of amygdalites. But amygdala, as we all know by the different talks we had before this, is connected to different parts of the brain. And how that it is connected with different parts of the brain is actually prefrontal cortex, ventrolateral, EEG, and then some other areas that I will talk about. But before that, how we can study fear memories, when we have to study the fear memories and the strength of fear memories, how we can, first of all, I would like to get you familiar with the animal model that is used for the study of fear memory. So what happens is that rodents are habituated in a box for day one, and then they are conditioned, and these are similar experiments. You all have studied physiology, Pavlov experiments. The ideology is like that. So here the conditioned stimulus is an auditory tone, is presented with the help of speaker, which is actually installed in the box. And at the end of auditory tone, you give a foot shock. So this is basically unconditioned stimulus, but there is an association between the conditioned stimulus and unconditioned stimulus. By this, after like a couple of trials, animal learns that this auditory tone is reversive and it is going to bring a foot shock for this animal. So after 2 or 3 trials, animal starts and learns that there is reversive event going to happen, and animal shows freezing response. So when you only give this conditioned stimulus, which is an auditory tone for a duration of 20 to 30 seconds, so animal shows freezing response. So that freezing response is basically a representative of how strong the memory animal has. So more is the freezing, more is the fear memory animal has developed. So this is a basic model. So if I can show you the video, I don't know whether it will play or not, that was made in the lab. If it is possible, it will be possible. I just wanted to show the video how like freezing is so clear that you can easily distinguish that animal is under the fear. I wanted to give a real look to the way how it looks like. So anyways, so aversive experiences powerfully regulate the memory formation, and memory strength is proportional to the intensity of these experiences. So the strength of memory is basically controlled by the circuit, and if there are certain inhibitions during those aversive events, then the strength of memory is defined. So for example, in these instructive pathways, which are responsible for the conversion of aversive signals, they are inhibited at certain points from ventral to RVM, post-ventromedial medulla. So then they set 2 conditions. Aversive event happened, but it was unpredicted. So the strong memory will be formed. On the other hand, like the aversive event happened, but it was predicted. Memory formation was strong, but not as strong as in the above condition. Like मैं थोड़ी सी मिसाल देना चाहूँगा ये कि आप गली में जा रहे हो, शाम हो रात हो, एकदम से अगर एक कुत्ता आता है वो पहले थोड़ी देर घूमता है, फिर आप पे अटैक करता है, तो आपकी एक aversive memory बन जाएगी. लेकिन if the dog appears and all of a sudden without like किसी को आपको चांस देने की आप पे अटैक कर देता है, that memory will be very strong. Because आपने उससे पहले सेंसरी जो सर्किट था वो आपसे स्टिमुलेट या इनहिबिट नहीं कर सके, and that resulted in a very strong memory. How that happens? So how is neural circuits construct predictive? इसको predictive error coding कहते हैं. So predictive error signaling during aversive memories, and in this mechanism controlling the strength of associative fear memories. So if you look at the circuit, the amygdala is shown here, and the foot shock is basically following this one, this somatosensory, and the tone which is coming, which is actually the associative fear memory will be formed, which is basically actually listen to the ears and to the auditory thalamus, and they are going to merge on the amygdala, lateral amygdala, and from here to the central amygdala, and from central amygdala to the periapical gray area. So basically, this is the circuit which is very important that I elaborated. That from lateral amygdala, the activity of lateral amygdala neuron is responsible for the formation of fear memories. And from lateral amygdala to the central amygdala, then there is the periapical gray area which is responsible for the freezing. And if we inhibit this circuit from central amygdala to the gray area, then the memory formation is very strong. So the same circuit is shown over here, where the negative color arrow is indicating negative feedback. So this is how actually the experimental setup was. The virus or archaeodoxin, which are activated by green light, they were injected into the central amygdala, and then they traveled, and then the ventrolateral PAG was inhibited, while at the same time the lateral amygdala neurons, they were recorded. And the infection of these cells in the ventrolateral PAG and central amygdala is shown here, that it was like specific infection in those areas. And then the training was done. What were three different protocols? They were low tone, it means like the unpredicted shock, foot shock was given, and then there was a predicted shock, and then there was a predicted shock with laser light on during that time, and this circuit was inhibited. So I will see this because this explains the methodology of regulatory options, because so what was found that during the unpredicted foot shock, the neural response is high, and during the predicted, this is predicted shock, foot shock, that the neural response is low. And when during the predicted and laser light is turned on to inhibit the neurons, once again the neural response increases. This is this graph here. On the y-axis you see spikes per second, and on the x-axis is basically time in seconds. And during the foot shock, the neurons were recorded and they were plotted here. So you can see that when there is a predicted foot shock, but we inhibit with the help of laser light, so the response is more like the unpredicted situation. So that's why the memory formation is quite strong. And this was proven in the behavioral experiments. How these were done? So animals were trained in day 1 in foot and shock association. 12 sessions were done, and then there was test 1, which is shown here. And then on day 3, again training session 2 was done, where the animals, they were divided into 2 groups. One group received the same number of less foot shocks and all, meaning the training was less strong training. And here in this case, the training was very strong, as you can see that there were 12 trials. In those where the strong or over training was done, in the test 2 they performed better, their memory was stronger as compared to the test 1 and test 2, the memory test 2 in the stronger foot shocks, that increased. So what we did was that, so this is the whole scheme where there was GFP was controlled and offset was the control when the light was given at some other time, and then the overlap is basically the laser light is gone. And in this case, you can see, although here is the model animal training model is very much similar. Here you can see day 1 training and day 3 training is same, but you can see that the overlap in case when the neurons are inhibited, the memory formation is much stronger in those animals as compared to the other controls where there is GFP and offset control. And this was further proven with the help of injection of NUST, which is inhibitor for the GABA cells, thus showing that that pathway is working. So learning strength is regulated by the feedback circuit of central amygdala to ventrolateral PAG. So here were some other experiments done where other parts of the PAG were also studied, but this effect was particularly for the central amygdala to ventrolateral PAG area. So I'm going to skip this. This is just the some additional experiments where the lateral amygdala neurons and PAG neurons, they were recorded during the behavior. So then from ventrolateral PAG, what is the next brain area where these neurons, they are coming from or going from? So what was done based on several experiments, a retrograde CTD tracer was injected into the RBN, RVM stands for rostroventricular medulla, is connected to the VLBAG. So it means when you inject here, those retrograde tracer basically tells it is those neurons which are connected with the VLBAG. These tracer will travel backside and it will actually show the staining in VLBAG. So how you confirm this? So when, like after doing the training, so there were once again box control, immediate shock, and then training. This is the actual urban experimental group. So we tried to find out those neurons which are FOS immunoreactive and CTV positive. Means FOS is standing for the FOS stands when there is neural activity. It means that neuronal activity was there in the VAG. And these neurons were basically connected with the RVM. How? Because we injected CTV tracer, which is retrograde tracer, and the co-staining of these cells is, you can see that, increase in the training. It means that the activity is high in those neurons. So this was further confirmed in the behavior. I will not talk much about this part of the slide because it is almost like same experiments repeated. But once again, training, same experimental protocol. Here, inhibition was done in the FOS staining, and inhibition was done with the help of RST. And you can see here when the light was shining on the VLBAG neurons. Once again, the fear memory formation is stronger. Although, the training protocol is same like day 1 and day 2, right? So this shows that there is connection from the central amygdala to VLBAG, and then there is connection from those neurons that are connected with the RVM. They are inhibited and they define the memory strength. So that was part of some experiments that were done. So learning strength is basically regulated by the circuit that was shown here from the central amygdala to VLBAG, and from the VLBAG to the RVM, and from RVM to trigeminal area, like that, the circuit shown here. So it is possible that, what are the applications of this study? So basically it is possible that the distribution of this feedback circuit in humans could lead to the heightened persistent memory, fear memories, and leading to the anxiety or post-traumatic stress disorders. So this study offers hope for the treating people with the elevated anxiety. And that's it mostly about my talk. And we have talked a lot about the aversive memories, so some soft memories. Wherever I usually give talk on the international forum, I used to keep 1 or 2 slides about Pakistan and want to give some image. Although this might be, you might be familiar that, but once again I would like to show that this is a unique place in the world, located in mountain areas in Pakistan. So what is uniqueness? Hindu Kush, Karakoram, and Himalayas. These 3 जो सबसे बड़े पहाड़ी सिलसिले हैं, वो यहाँ पे आके मिलते हैं. And this is one of the unique places in Pakistan. So let's live. This is my message. Let's live together, happy life, and make our planet peaceful planet. Thank you so much. If there are any questions, I am here today and tomorrow we can take those questions. And thank you for listening patiently.